Hi, everyone. Thank you so much for joining us today. Before we officially begin, I do want to point out to the audience that we welcome questions. So please post them in the Q&A section at the lower part of your screen, and we'll address those questions at the conclusion. So I'd like to now turn this over to Stephen Saltzstein, Forrest Family Office's CEO.
Thank you, Callie, and thank you everyone for joining us today. We have a fantastic discussion with Bernard Pepperstraat, CEO of AcuamarkDX. Bernard, just to begin with, can you tell us a little bit about AcuamarkDX, what you guys do, what you're targeting, etc.?
Sure. Thank you, Stephen. Thanks for having me. Thank you for inviting me. It's an honor to see you again and to be here again.
Honor's mine.
So AcuamarkDX has been focused for a while now on early cancer detection technology. So we have built technology that for the first time has now allowed us to detect cancer, stage 1 cancer from blood accurately. So you have seen a lot of other companies in this space that in the diagnostic space that have tried to target early cancer detection. But we've seen really modest rates of success when it came to really nailing the problem of stage 1 detection. And the reason stage 1 detection is important is obviously that's where most lives get saved. If you look at colorectal cancer, for instance, colorectal cancer goes up, the survival rates go up to 90% if one would reliably detect it in stage 1.
Yeah, great. And so how big is this opportunity? Can you talk about the total addressable market?
Yeah, so this is just CRC, of course, or colorectal cancer across Europe, US, Japan, South Korea. We count the need of about 175 million panels annually. That's assuming that we would get 80% compliance. Currently, the compliance for CRC screening stands at 60%. And of course, that's assuming that we would do a biannual test. So 175 million panels, which equates roughly at CPT code pricing that we currently see in the marketplace, which the CPT codes that would apply to us, which would equate to about $100 billion market.
We were just talking about comparables. Can you kind of just give us the lay of the land, the 2 or 3 other major players that are out there, how you stack up and their valuations, etc.?
Yeah, the closest comps really are Natera, because we also pursuing colorectal cancer recurrence. Natera at $21 billion market cap today. That might have been a couple of days ago, but they sort of fluctuate around $20 billion. Exact Sciences is the second one, which is famous because it has just been marketing Cologuard for some time, and now recently came out with Cologuard Plus, which frankly is a very good stool test. They hover around the $10 billion mark in market cap. We saw Tempus come out of pretty much nowhere in the past few years with a heavy AI sort of fueled approach. And then we see Guardant Health as well. Guardant is into this game as well with a recurrence test and also with their early detection test. Guardant is maturing. It's hovering around the $5 billion market cap. The interesting piece about both Exact and Guardant was that both public companies, when they went public, the market really received them very well. And they achieved revenue multiples. In the case of Guardant, that was 78 times, I believe, revenue multiple 4 weeks after they went public. And that was not dissimilar from the time that Exact Sciences went public. That was over a decade ago. And so we see that typically these companies get very good revenue multiples going for them.
And in the case of Exact, right, Cologuard, been public for over a decade, never been profitable. Can you talk about why you guys are going to be different and what differentiates you from the competition?
Well, one is to imagine it's a stool test. And so we've seen, I'm going to start from the top down in a way with the reasoning, but stool tests can be taken from the comfort of one's home, right? But we've seen that unit sales wise, we saw 20 million tests being sold roughly over the lifetime of Exact. And if you look at the marketplace need is 40 million people who do not get CRC screenings in the US alone. And so if one looks at that average a year, roughly average a rough average would be 1 million tests a year or so. That really doesn't make a dent in that need of 40 million people that do not get addressed, that compliance gap that we see in the US for CRC screening. And so we think that a blood test that comes within striking distance of some of these gold standard performances, but is easily administrable and prescribable at your GP's office eventually, right? Can be taken right next to your lipids, right next to the other screening tests that you get in your annual blood works. That will allow us to really quickly, fill that compliance gap more easily than with a relatively cumbersome stool test. And so of course the approach that we have developed is one that was not just innovation, it was a slightly higher bar. The innovation was within guardrails. And the guardrails were, how do we do this? How do we build a very accurate technology, but that also uses equipment that's already commercially available, uses the workhorse of the industry, uses high scale, high throughput type of equipment so that we don't have to reinvent the wheel in terms of equipment. And that equipment is called qPCR. It just wasn't used yet, or it wasn't possible to use it yet for complex biological problems such as cancer. So we had to build assays specifically and innovate on assays specifically to allow us to look at the type of markers that would apply in our view for cancer, but secondly would allow the assays and the technology to be broadened in such a way that it could look at complexity of cancer. And that then is mounted on universally present qPCR equipment. It's, people recognize the terminology of qPCR because of the COVID crisis, right? That's when the most reliable qPCR COVID testing was done on qPCR. It's used around the world. And therefore we thought, well, this approach, if we can build something that's really that accurate, that reliable, but can still be accessible via universally accessible, commercially available equipment, and by the way, has low margins, right? Has high margins, low cost of goods, that would be a winner for everybody involved. And so especially the patients. And so that's where we think we make a difference.
Let me just understand. I just want to make sure everyone understands. So you get approval, and then how do you get distribution? You kind of like touched on it a little bit, but like, how are you going to get integrated into the existing platforms?
So we trying to be as seamlessly fitting with current workflows as possible. And that means the workflow that we've developed fits seamlessly with, or should fit seamlessly with workflows in current laboratories, like commercial laboratories. Secondly, in terms of the clinical physician workflow, to physician's office, nurses, etc., it's just a qPCR-based prescription. It's just a prescription such as the COVID test, such as your lipids, etc. So it's designed to be seamlessly and frictionless, right? And secondly, from the biological point of view, we wanted to be able to retest individuals if it would be needed, retest borderline cases 3 months down the line, 2 months down the line were needed, right? Because you cannot imagine that happens with an elaborate, expensive, long turnaround type of next-gen sequencing test. It's also far more challenging to imagine that would be happening with a stool test, for instance. So, but just because it's a qPCR, straightforward qPCR prescription test, just because it's a fast turnaround test, it's accurate, easily done. One can easily ask individuals to come back 3 months down the line and then get to even higher sensitivity specificities. Why? Because 3 months down the line, for those that are borderline cases, we would shave off even more false positives, shave off even more false negatives, etc., and hence get an even higher sensitivity and specificity than we currently get.
Let me ask one more question. I do want to go into why people need to do this early, etc. But who else has a blood test out there?
So we've seen Guardant Health has one. Everybody might have, many have heard of Grail, of course. Grail has a blood test. We see Freenome has a blood test. And then a slew of other companies that are still private, like Delphi, Adela, etc., have blood tests as well.
And how does your efficacy stack up against them?
Yeah, so very, very well. I mean, as a matter of fact, it's in stage 1 and stage 2 that we are pulling away from the pack. So the latest tweak of the assay, the latest version of the assay, and that's just hot off the needle, really, just months old. These data are giving us a detection rate or sensitivity of 88 to 100% in stage 1 and 90 to 100%, 90 to 100% in stage 2. And the reason there is a range is because we applying machine learning AI to this, and so each of the machine learning models spits out a slightly different sensitivity, but the specificity remains the same throughout, right? So a positive is a positive, and so that for that reason, the specificity that we see is at 97% for the best models, which is really where this becomes a very exciting test. We have very low false positives, so it's very good for the payers. We have very accurate rates in stage 1 and stage 2, which is very good for providers and patients.
So talk to me about survival rates with stage 1 detection, stage 2 detection. Like, why is this so important?
So generally, and this is true for most cancers, right? But this is very pronounced, for instance, in CRC, colorectal cancer, which is our first product that we're going after. In colorectal cancer, in stage 1, survival rate goes up to 90%. Stage 2, survival rate remains 70%. Stage 3 and beyond, it falls below 50%. Gradually, more and more and more. It metastasizes 10%, right? And so that's where it lies. So today, in aggregate, if you look at all the cancers, across the planet, we have a survival rate of cancer about 50%. And that's not CRC, that's all cancers in aggregate. And that's roughly tied to a detection stage that's in most cases at stage 3 or beyond. And that's just so the gap that's not solved is the stage 1, stage 2 detection gap. If one can own the stage 1 space, it's really where things get very exciting for patients.
Is this a platform technology? What are the other cancers that this could be applied to?
So that's a great question. I mean, we are a platform in several ways. We sort of try to, in a very orthogonal fashion, try to make it a platform. So we are a platform in the first that we built new assays that fit on qPCR as a platform itself, and that allow us to really sample methylations for the first time easily from blood. Sample low abundance, ultra low abundance methylation for the first time easily from blood because it's qPCR. So that's the first level of platform. Second level of platform, this means we are a methylation platform as well. Not just an assay or qPCR platform, but a methylation platform as well. Methylation means it applies to a great amount of aging-related diseases. And cancer being an age-related disease, within the cancer group, of course, methylation remains highly relevant. And so the way that we're going at this is we're starting with CRC, and we'll add gastric and esophageal to it. That becomes a GI screening test. So this goes then beyond the discussion of stool testing, of course, very quickly, right? And then we go into the next family of cancers, which is lung. We expand within the lung family as well. And then we move to women's cancers. And so the pipeline is really stacked to first and foremost focus on and tackle CRC. But then once we've nailed CRC to really with very fast cadence, start working on the other broader applications within the same families, add lung, add women's cancers as well, etc. And so what you will see in the future is hopefully we'll get there, right? But if once we launch CRC, we'll be able to bring on the other cancers again, fast cadence. Time between each of these launches should be about 1 to 2 years.
So, what's the commonality in regards to your IP and your just kind of know-how that maybe hasn't been patented that allows you to use this platform for many cancers? Can you talk about kind of the detection of action, so to speak?
Oh yeah, totally. So if you would, in a way, if what would tell you a lot is the chronology with which we've built the IP and the patents. And so if you go back all the way, you will see, or you would see, that the first intellectual property families are all methods based. So foundational new methods from the ground up. And we got through international filings without really much pushback. So this was a relatively wide open space. It's very good for us. About 8 families later, the last family is now laying on my desk. And between those very broad methods patents and the latest one, we have patents that get more narrowly focused on products. So the closer we got to product, the more narrowly focused these patents became based on molecular structures, primers, probes, etc., which start having the strength of a compositional matter type strength. And then the latest patent is really trying to encapsulate the work that we've been doing, uh, uh, in collaboration with the collaborations that we've been doing between the assay folks and the bioinformatics folks, and we're trying to really encapsulate it in the last family of patents. But then there is, of course, also a component. There is some things that are not protectable in the US and Europe, which are facts of nature. And so there's many facts of nature, such as is a piece of DNA is methylated or not methylated. That's not something we can protect. That's a fact of
Right.
nature.
Sure.
So the tweaks with which we do some of that are covered under trade secret. So
Got
it covers the whole gamut, basically.
Right. Can we talk about just the healthcare system in regards to is the platform cost savings? What is the systematic impact that the platform can offer?
Yeah, so that's again an amazing question. Thank you for asking it. So what drives us is, of course, right, getting this to the patient quickly and allowing us to save lives, right? Because at the end of the day, if a technology like this would be deployed, we could save up to 50 to 75% of lives that currently die from cancer, right? So that's number one. Number two, that's on the patient and provider side. On the payer side, and this goes beyond just medical payment system or reimbursement, and this goes beyond insurances, all of that. The WHO estimates that the cost of cancer, and that's broad definition cost, that's productivity loss, that is anxiety caused treatments, that is people putting their houses up for mortgage because they need to pay for treatment, etc. That kind of cost is $1.2 trillion across the globe. In the US, we count at about $400 billion a year. And so if one shifts the detection to early detection testing, with all the pros and cons, all the cost that brings, but also all the savings that brings, we should be able to save about up to $300 billion in the best case scenario, high compliance and all of that. But the potential for that is to save $300 billion out of the 400 billion, right? And that's just simply by detecting cancer at early stage 1, where treatment is far simpler, where there may still be a need to have follow-up chemotherapy, but definitely far more modest, and in some cases not at all after surgery, right? And so it is of course a far more nimble treatment flow that we see, but also beyond that there is great opportunity to benefit society in terms of reducing anxiety levels associated with detection of cancer, reducing the impact on families that it has, reducing the economical devastating impact, not just of treatments, which can go up to several hundreds of thousands of dollars, right? And sometimes more, millions of dollars if it comes to some of the experimental treatments, and sort of reduce that aspect as well. And so
It's
that's why we
funny.
get really passionate about it.
People, obviously the big debate in the US right now is about the big beautiful bill. And part of the rationale that's being claimed about it is that we're going to see revenue growth from AI, right? AI, the revenue growth is going to take care of the debt burden. And it's interesting to think about, like, if we were to just reduce or just have citizens focus on early detection of cancer, how much money that would put back into the system.
Yeah.
I think that's nobody's talking about that, but I think that's pretty fascinating, actually.
It is actually we consider it an asset for the approach almost, and so it's an asset that's never being discussed. But if you take comparables with events, there has been lots of damage created for relatively little savings. Not to get into the politics of it all, but the
Right.
thing is, with just, if you were to look at a technology like ours, how much would it take to get to public markets and put this thing on the market and bring it to the patient? Probably 125, $150 million. And what would be saved? Well, there you go, $300 billion in the US.
Right.
Now that's the savings.
Yeah.
So we think there is we just haven't had lots of visibility yet, but we're getting there. It's time for us to ramp up and to stop bootstrapping ourselves now. We are the crossroads with the company. We've established a clear demonstration of the performance of the assays, and now for us it's time to really step things up, and that's why we're doing the Series B raise, of course. But to your point, yes, the cost versus benefit ratio in our case is astronomical and fairly unmatched with many of the discussions that are being held at a bureaucracy level. And nobody likes bureaucracy, and everybody wants to see it reduce and be more efficient. But technology can indeed, to your point, technology can indeed greatly improve economical situation in society. And innovation in our case, which has been a long road, and a tough road, but innovation, which is not easily funded in the US, right? True innovation, it's not easily funded in the US, even less so in the rest of the world, is something that can really make a dent very easily, in these huge budgets that we see.
Have you guys had to overcome kind of major bureaucratic hurdles like reimbursement, infrastructure, physician adoption, things of that nature?
Well, we're not there yet. So we are test and development, and it would take us about 18 months to get to commercial system lock ready from here. Like after Series B closing, it will be 18 months to get to commercial system.
So is there a code already for this though?
We have identified CPT codes,
Okay.
which we could apply directly. And so
Okay.
a commercial system lock implies, number one, we could start an FDA pivotal study. Number two, we could, in principle, launch with a limited launch, and for instance, direct payment agencies or institutions, think firefighters, think individuals that would need this and that would be happy to pay directly for it. That would be an immediate avenue to get that to them. This would be at the same time we're doing the FDA regulatory studies to get IVD approval. But so it gets us commercial ready, and because we're planning to add to bring the 350 study sample set to about 3,000 at the point, we'd be M&A ready as well. About 18 months after the Series B closes. Why? Because we've discussed this with quite a slew of large companies.
And so what does that mean? Does that mean that, okay, once you get FDA approval, you're then gonna partner this with large pharma, license it out? Like what's the theory there?
Well, actually, before we start the study, we'd be M&A ready as a first point of M&A readiness. Why? Because I like optionality, right? We don't have to do it, but we'll be a bolt-on type of solution. And if people want to start being pragmatic about it, we'll be pragmatic about it. Why? Our North Star is let's get this to the patient as fast as we can. And so if there's institutions that have great sales forces available, why would we reinvent that wheel, right? So we'll be pragmatic about it. On the other hand, we will not leave money on the table, right? And so for that reason, we just want to say, well, we will be M&A ready as an optionality exit point, but that we'll keep preparing for the IPO route. Why? Because in the public markets, we see very good multiples, maybe better so than M&A, right? But in any case, so that's sort of the path that we carving out for ourselves a little bit. One of the themes is optionality, and so that's where we think this opportunity from the investor point of view and is.
Listen, I think that what you're showing everyone right now is you're a very good CEO. And I have very few conversations like this where people are so focused on kind of you just seem to have a very well thought out roadmap. And I think where a lot of management teams fail, especially in life sciences, is they can't get from science to business execution. But it's very obvious to me that you have meticulously thought this out and have a good strategy.
Thank you, Steve. Well, look, the job comes with having to think 10 years ahead, 5 years ahead, of course, immediate ahead as well. But then again, when I started this company together with my co-founders, so Franz Barani, co-founder, relatively notorious and renowned inventor, and David Gelfand, inventor of qPCR, co-founder, and all of that. I decided to do this even though it was just still at conceptual stage, no data. We're just going at it because we believed new assays were needed to really nail this problem. And I figured, and I think everybody joined me in figuring that this need is so vast and this technology could be so powerful that we don't have to be concerned about the money following or the economics following. We just have to nail stage 1 detection. We had to build an entire new assay from the ground up, and we're the only company in the industry that has done that. But and that comes with risks, and it comes with lots of troubleshooting. That comes with 1 step forward, 4 steps back. But it comes with eventually just a really hardcore focus on getting that stage 1 detection nailed. And so I think we have now demonstrated that our assays could do that, and they do exactly as we hoped they would do.
Can you talk about your existing team, but really the broader team that you will have to build out if valuation doesn't match up with your expectations around lab commercialization, et cetera, market access?
So yeah, I mean, I see you listen carefully. We are at a crossroads. At this moment, anybody can basically say, well, why don't we put it in our lab, stabilize the assays here for commercial system lock, and do it ourselves? So that's all true. And so what we're currently planning to do, of course, with our Series B raise, is expand the team, and that will be on several fronts. Currently, in the past, we've been focused on pretty much the assay development in the teams in the labs at Weill Cornell Medicine. Going forward, we'll need commercial system lock. What that means is we'll need our own little lab footprint. What this means, we'll need a number of technicians. We've mapped it out very carefully in our budgets as to what cadence they need to be attracted. We'll also need, now some more senior leadership to join me, and we've identified some people that would make this an absolute killer team. And then we'll need to think about, well, at that 18 months post Series B closure, 18 months afterwards, do we or do we not go in parallel to the FDA study? Do we or do we not go commercial as an LDT? It's a limited launch. And so if we do, and that's a business decision, that's depending on our margins, which are of course very good on because it's a qPCR based test. But it's still a decision by the board and by investors. They must want to do the expense, and the expense looks as follows. We would, if we do decide to go commercial as an LDT as well, while we're doing the FDA study, we would start adding product specialists a year before launch. These product specialists would start educating the market. The market involves, the typical strategies in life sciences, which is approaching KOLs, educating them, let them do presentations, etc. And then those product specialists would turn into reps. We'd grow the marketing and sales force that way. Now, question remains: is this something that we need to do, or should we just have somebody else do this? We will probably, if we decide to go forward in LDT, we'll probably do that ourselves because the cost that we would have to pay somebody else to do it would be high. If somebody decides, well, this is something we can easily bolt on to our company, and we know some large companies that will need this, we think, then they of course have the sales force in place to pursue this. And so from here on, it's just optionality / pragmatism that will determine what we do, but also sound business decisions.
Last question before we get to the audience questions, which is, so how much capital have you raised so far, and who's backed you, and what's been the use of proceeds thus far?
Thus far, we have done 25 million raised. This was over a number of seed rounds, so 3 seed rounds and 2 A rounds. The investors are pretty much predominantly angels and large family offices. And then we have one venture capital firm from the West Coast. The reason they invested was because the principal had been involved previously with both Natera and with Garden Health, and so he really liked the story. And then we also have one corporate, nothing, no strings attached, just a minority investment from a large corporate.
Okay, super. All right, we have a bunch of audience questions, so I'm gonna let Callie take it away. The floor is yours.
Okay, great, thank you. So what specific biomarkers or technologies enable you to detect early stage cancers with high sensitivity and specificity? And how do these differ from competitors like Grail or Freenome?
So we started very early on with a broad sort of exploration bioinformatics-wise. We thought what would be needed would be mutations plus, methylations plus, maybe even proteomics plus, markers called messenger RNA. And we quickly learned, based on the way we looked at these bioinformatics databases, that our marker of choice would be methylations. And then later on, we saw that both Grail, Freenome, etc., also moved to methylations. Guardant is using methylations as well, but is also using fragmentomics, etc., etc. But the differentiator is not the type of marker specifically only. Of course, carefully selecting the marker panels is essential. We learned that we would need to get a very clean picture, not one that has too much noise in it, but not one that has too few data points in it that is not a picture, right? But to have a good enough picture, a clear enough picture, we would need about 40 markers to be tested. qPCR couldn't do that. qPCR couldn't do 40 markers, and qPCR could certainly not do methylation markers. And so it's almost a result of subtraction, right? Whereby it's not 10 markers, like the older approaches, such as you see sometimes with companies such as Exact Sciences, for instance. You have a ceiling with 10 markers. It's too limited a data pool to really learn enough about the broad heterogeneity of cancer that cancer presents us with. That's on one side of the spectrum. On the other side of the spectrum, you have next-gen sequencing companies, Freenome, Guardant, Grail, etc., that really have used next-gen sequencing, which is millions of markers. And so the next-gen sequencing technology we think is very elegant. We don't want to replace it at all, by the way, and I'll explain later why. But the analyzing a million markers is creating a lot of noise. So while you're trying to look for the needle in the haystack, you're also creating your hay. And so and that's why the results sort of fall off the cliff when it comes to stage 1 and stage 2 detection for Guardant, Grail, Freenome, etc. As a matter of fact, we see their sensitivities or detection rates for stage 1 linger around 50-60%, while ours is at 88 to 100%. And so that getting that right amount, not too little, not too few, and not too many markers analyzed, and of course have the right selection of markers, then of course we had to enable qPCR to do that, is what really allowed us to do this. And we had the help of AI and machine learning. And so these novel assays, together with the second pillar or leg of the stool, which is the selection of markers, together with the AI machine learning as a third pillar or a third leg of the stool, is what altogether is what makes the results the way they are.
Great, thank you. What is your anticipated regulatory pathway, and what milestones must be achieved in the next 12 to 24 months to reach commercial development?
Great question. So we have our regulatory consultants lined up, so they are very clear in the path that we have to follow. And so for the CRC screening test, we'll have to follow whatever Exact Sciences is doing. What that means is we have to do a PMA study. PMA study is, once we are ready to start a PMA study, that will be a 12,000 individual study. For us, we don't think it's really a big deal. It's 12,000 samples to be taken. It's not like follow-up visits and a therapeutic study, but it's still a sizable study. At the same time, we will be able to sell as an LDT. In terms of market access strategy, we'll be able to launch already as a lab development test. That's one path that exists for diagnostics companies. You don't have to have regulatory approval per se to launch as an LDT, but it is more limited, more restricted. So we don't expect large amount of revenue, not like hundreds of millions of revenue under an LDT. We expect 5, 10, 15, 20 million, enough to build our presence in the market space a little bit. And then once we get approved, this can take off. Now, in terms of CRC recurrence, so the recurrence test, as the one you've seen at Guardant Health, for instance, or at Natera, the recurrence test, the regulatory path is a 510K, and the 510K just is a sample, is a trial of about 450 people. So obviously the development cycle and the market access path is far shorter and far more doable. But in our case, both the screening and the recurrence test have strong precedent. And then market access wise, CPT coding, right? CPT coding that we have identified for the screening test is about $592, $94, sorry. So $594 for screening test, which is the CPT coding for Cologuard Plus. So we would just adopt the same one initially. And then for the CRC recurrence test, the CPT codes that we have identified would provide us $392 times 4 a year, because obviously once one has been diagnosed and one monitors for recurrence, it requires several monitoring visits, right? So it's 4 times a year, $300+. And at cost of goods that we project this to go down to about $40, $50 cost of goods, we think the margins will remain very healthy. And that's one other differentiator that we see from lots of the other companies that run pretty much on negative margins. You see in many cases their revenues ballooning. You see their market caps ballooning, but you see also their net losses ballooning. And so we don't think that's very sustainable. And as a matter of fact, the reason I like to keep profitability as one of the foci of what we do is because if we eventually go into innovate, not just on CRC, but on gastric esophageal as well, on lung as well, on women's cancer as well, we'll need profit margins in order to support it R&D.
Great.
Or free cash flow in order to support it R&D.
So how do you plan to integrate your test into routine care settings? Are you targeting primary care networks, health systems, or DTC channels like telemedicine or retail clinics?
So there is a bit of a crossroads that is ahead of us. If we decide to do LDT, then we will rely on direct pay for a little while, until like 6 months, a year, until we have the CPT codes assimilated into our approach, right? But that would be what we foresee is then potentially access to direct pay type of institutions. But eventually, this ought to be a regulatory approved IVD test that's available and reimbursed as any other tests, just like the Exact Sciences tests are being reimbursed. And that means we'll just have to do the FDA path while we're already commercial, of course, but we have to do the FDA path and then subsequently follow reimbursement studies and outcome studies that the insurers and Medicare and Medicaid wants, depending screening recurrence. In terms of getting this into clinics, will eventually be a test that's sold to insurers, right? And so physician practices will just be able to order this and prescribe this, and hopefully, will be available through insurers at that point.
Great. What key hires or partnerships, clinical, commercial, or regulatory, are needed to de-risk the execution in the near term?
It's a great question. I think team-wise, I mean, and this is well laid out in the budget. Again, we've carefully calculated which team members we need when, but team-wise, we need help. We definitely need senior leadership, and that's on the operations/lab front. Regulatory-wise, we have the right blue chip consultants that everybody else uses as well lined up, but we'll need internal staffing for that as well. And that's both regulatory market access. On the lab operations, our own lab footprint will need technicians, but also lab directors, quality assurance people, etc. So team is definitely one. In terms of partnering, so there would be a great number of parties, and we are talking to we talking to several other parties with in mind sort of a strategic angle, and that strategic angle can be the ones we like most at the moment are, of course, the ones that provide us both early exposure to the technology as well as samples, right? So what I'm talking about is partnering up with some of the most well-known healthcare systems in the US. We have started one collaboration with a large system in Latin America, one of the highest regarded ones, but we want to expand that into the US with 3 to 4 large partnerships that would get us access to samples, but also that get us the buy-in from the PIs and the KOLs at these institutions. So far, the discussions we've had were without exception incredibly positive with each of these KOLs. And so we get the strong buy-in with some of the who's who in in the US from these institutions. Corporate-wise, that's a bit of a open-ended sort of question, right? We don't have anything on the table at this moment, nor do I know whether we will have anything on the table. But corporate-wise, I think we at the moment, if we're going to make this commercial system lock ready, what that means is also we have a pretty good workflow at the moment, but we'll have to now make a choice. If we bring this into our own laboratory, we'll have to make the choice which instrumentation we will use in our workflow, which sets of instrumentation automation we will use. Why? Because if we want to do an FDA study, the FDA will want to know the serial number of the instruments that we use, right? So it must be fixed. And so that's something that is, that once the Series B closes, we'll be paying very careful attention to. And that might behoove us to just have that done in partnership with one of these instrumentation makers.
Thank you. What is your go-to market pricing strategy, and how does your cost structure allow for broad adoption?
Yeah, so it's a good question, right? We try to, and I keep repeating this, I guess, but we try to not reinvent the wheel, and that means is part of the reason we went into CRC, for instance, is not just because it is biologically a well-established cancer, not just because it's a technology we know, it's a cancer we know well, but also because it's a well-established market, right? It's got lots of precedent, regulatory-wise, as well as market-wise. And so for that reason, we have CPT codes we can use. So market access strategy, CPT coding is, again, it's $592 or $594 per test for screening tests. And then we would decide whether we want to go to CMS to ask for our own codes later. It takes about 2 years to do that, but immediately we could start assimilating the CPT code that Cologuard has. And then secondly, the other relevant one is for CRC recurrence, and that's the 4 times a year, $394, I believe, right? Cost, and then put that on a cost of goods of about $40, $50, which in commercial system lock, we should get it down to that. The margins, of course, are healthy that way.
Great, thank you. Um, and looking out in the next 24 months, how much additional capital are you thinking that you need to raise? And, what would that unlock
Yeah, so
between launch, revenue, studies?
yeah, so from here on, it was actually quite simple. So we've done the hard work, and from here onwards, we need to achieve two things. One is commercial system lock, so that we are at the point where we can start a pivotal study. We'll be at a point whereby we can start thinking of commercializing, and we'll be M&A ready as a first point of optionality, like just the first point of where we M&A ready. That could be within 18 months, but say 18 to 24 months of Series B closure. The Series B is designed to cover for a commercial system lock, and secondly, to bring our sample sets to about 3,000 samples. 3,000 samples is a case control type of design, 1,000 cases, 2,000 controls, are the numbers that we kept on hearing from some of the large companies we've spoken to. We've spoken to 3 in particular, all of which came back with that league of sample data that they would need to really look much deeper, which is why I say that's the first point of optionally be M&A ready, but we'll have 3,000 samples under our belt at that point. It's also the that's also the amount of samples, coincidentally, that we just saw coming out from Cologuard's, the Exact Sciences blood-based test, where they reported on their performance for colorectal cancer, which wasn't great. And so we'll have a very good idea at that point based on that sample set, and our additional data, whether we will have a Exact Sciences-like company by the tail, or an Exact Sciences better company by the tail, or a Natera-like company by the tail. And you look at these market caps, $10 billion Exact Sciences, $20 billion Natera. I'm not saying this will be us. I'm not saying that point will be valued that way. But at the very least, we'll have a very clear idea whether we equivalent or better than these companies in certain ways. And so it will put us in a very powerful position about 18, 24 months after closure of the Series B. Will put us in a very powerful position to make the argument that we deserve a seat at that table.
So you had mentioned M&A. Can you outline, other potential exit scenarios, IPO, long-term private scaling with a partner?
Well, that's actually a very sophisticated question as well. I mean, especially the last piece. Very interesting. So there are typically, most people think about exits in 2 ways, M&A and IPO. So some people add a third one, which is stay private, hold a company in a limited amount of hands, and let the profits come in, right? And with this kind of company, with the margins that we see, the vastness of the market, the third scenario actually does make, in my mind, a lot of sense, right? But it requires the right partners and the right people that invest, because this means one does not rely on the capital intensity of the public markets, and one does not rely on potential cash that comes from M&A transactions, right? So but so be that as it may, to answer the question, so we recently added an individual to our board. He used to be at EY. You can easily look up who he is, but we did that because we wanted him to help guide us through the path for M&A readiness, but also to IPO. So it's a very senior member of EY. He has seen hundreds of these and prepared hundreds of companies for that. And so the idea is, given the multiples we can get from IPO is a very viable path for us. And so about 18 months after we're done with the proceeds of the Series B, meaning say the Series B closes today, 18 months from now, we would be M&A ready. 18 months after that, we would be IPO ready. So it's about 3 months. The comfort zone is about 3 months in terms of preparation for an IPO, to get IPO ready, and about $10 million price tag. So we have not put IPO preparation in our budget for the Series B, which is a $25 million budget. We've not put that in there, and for that we would need a Series C subsequent to the Series B. So the larger rounds following the Series B would cover IPO preparation and the budget for it, and would cover getting this to public markets and getting this scaled up in terms of marketing and sales.
Okay, great. Well, that concludes the questions. For the audience, anyone who has additional questions or wants to be connected, I'm happy to facilitate that. Everyone out there has my email address. In turn, Bernard will have all of your information as well and will be reaching out. Bernard, I'm going to turn it back over to you for any final words before we close out.
Well, I thought this was just a wonderful event. Thank you so much for having me. And then I also would like to thank the audience. The questions were just spot on and very sophisticated. So I really appreciate that. And if there are any other questions, I'm of course always on standby and available to answer any other questions. But thank you so much for making so much time for me.
Great. Thank you so much, everyone, and have a great day.